Hello friends. So in last lecture we ended up at eukaryotic cells was finished and we were about to start prokaryotic cells. So let's talk about it. So here we have generalized prokaryotic cells, in which you can see the major difference from the eukaryotes. It do not have a regular shaped nuclei, while its DNA is circulated in plasma. The first major difference. Then is the flagella. It is also present in some of the eukaryotes, but it's a major feature of prokaryotic cells. And you can find some organelles like ribosome, plasmid, and plasmids, which are non-membrane bound, but there is no mitochondria, no nucleus, and no other membrane-bounded organelles. These are the pili and everything. And coming to the another difference that I think I missed in the last lecture, that it has a cell wall. The outermost boundary is not cell membrane but a cell wall around cell membrane. This is the outermost layer. So these are the differences among these. Let's see what is written in the slide. Unicellular organism found in all environments. These includes bacteria and archaea. Okay, I discussed in the very beginning of the previous lecture that archaea and bacteria have prokaryotic cell without a nucleus, no nucleus, and no membrane bound organelles. All that I just explained. Cell contains only one circular DNA molecule contained in the cytoplasm. Okay, DNA is naked, no histone proteins. So this is the difference. The DNA is naked. This is not bound by the histone proteins which are present in eukaryotic cells, and cell division is simply binary fission. That one divides into two. Here you can see here is one cell, and it will just start making a groove inside at this place from both sides, and later on it will be divided into two different cells. Those two will completely new cells. Okay, coming back to archaea. Archaea is prokaryotes organism without nucleus, but some aspects of their molecular biology are more similar to those of eukaryotes. So that's why we put them in the separate category of archaea. Okay, so the next thing is genome, totality of genetic information of an organism encoded in the DNA, for some viruses in RNA. So genome is the genetic information of organism. The total entire genetic information inside an organism is called its genome. We can say in other words and all that genetical information which is encoded in the DNA. So all the DNA and all the information that is in DNA is called a genome. And for viruses, some of the viruses have RNA, so it is saved in RNAs. Okay, the genome size. Human genome consists of 6 billion, the size, while plant is much more larger than this one, mammals. Then coming to fungi, which is 10 million, E. coli, bacteria, which is 4.7 million, and for MS2 its viruses it's 3200, very small, and viruses it has 1 lakh. So this is the size of genome in BP. It to 10 raised to the power of whatever. Here it's 10 raised to the power of 9 something. Special number of chromosomes. So we have to remember that all organisms have some specific number of chromosomes in their body, in their cells. Every cell have 46 chromosomes in humans, which means 23 pairs. Mouse have 20 pairs and 40 in total. Rat have 42. Fruit flies have 8, and bacteria have only 1, because it is not a prokaryote at all. Okay, human genome arranged on multiple chromosomes, 23 pair of chromosomes as I told you in the previous slide, 22 pairs autosomes, 1 pair is sex chrome, XX for female and XY for male. Here comes the gametes, so which are sex chromosomes, by 20 other pairs are autosomes. Humans have 23 pairs of chromosomes in very cell, except mature red blood cells. Gametes or sex cells, sperm and eggs have half the normal complement of chromosome. So that's why these are called haploids. Haploid. Yeah, these are haploids, while other 22 are diploids. So when the sperm eggs combines, it makes one diploid cell, which can later on divides to produce new cells, multiple copies. So here comes the genome, entire genome, in which this is one chromosome, this is the genome, if then this is the chromosome, and in this chromosome there are genes, and in between is the intergenic region. The genes have a specific information for some kind of trait, any of the trait, which will be transferred in the next generation. So we will end up in at this slide, which includes human genome have 3.2 GB nuclear genome, and mitochondrial genome is 16.5 KB. You can find the difference in the size. And genes related DNA are 25%, and extrogenic DNA is 75%. We're dividing the genes and gene related DNA into coding and regularity DNA and non-coding DNA. Non-coding DNA includes gene fragments, pseudogenes, introns, and untranslated sequences, while extrogenic DNA have unique low copy number DNA and repetitive DNA. Repetitive DNA further have two divisions, tandem repeats and interspersed repeats. While mitochondrial genome, which is 16.5 KB of size, which have two rRNA genes, 22 transfer RNA genes, and 13 polypeptide encoding genes. So this is the division of entire human genome in human. Now later we will study about structure of nucleic acids, but today's lecture ends up here. We'll be right back. See you.